Hello everyone, Dr.Mungli here. So in this video, I will be going over a very important concept that is repeatedly tested in all kinds of exam, whether it is USMLE, NBME, Kaplan exit entry exam or a next exam or any PG entrance exam. So let's get into that. Read the question that is appearing on the screen right now. You can pause and read the question. I'll come back in a moment. Alright, so we have read the question and there are multiple choices that are given here. So what is your answer? So in order to get the correct answer, all you need to do is to figure out what is the metabolic process that has been discussed in this question, what is the most important enzyme to begin that metabolic process, and what is the activator of that particular enzyme. These are the three things that you got to figure out. So this is the third order of question here. Okay, so some of the things that you really need to look into the question is this particular man has on a self-induced weight loss plan. So he has been fasting for 48 hours now, and one of the primary enzyme that is involved in maintenance of the blood glucose level coming from the liver and synthesizing glucose from non-carbohydrate. So before I go to the answer, I know you have your answer there. So before, let me explain the process. So whenever we go into fasting condition, so 3 to 4 hours after meals and then 12 hours and by 24 hours, it is the glycogen degradation that is going on in the liver will maintain blood glucose near normal levels. And then gluconeogenesis. So gluconeogenesis, although it begins 2 to 4 hours after meals, but it starts to steam up at 12 hours and then by 24 hours it will be at peak. I have a video on this. What are the different sources of glucose for the brain under fasting and starvation? The link for that video is in the description below. It is there appearing right now at the upper right corner. Anyway, so gluconeogenesis will take over glycogen degradation by 24 hours and it will be there for another 4 to 5 days and then it declines. You got to remember that. So here person is fasting for 48 hours now. So it's a gluconeogenesis. Where is gluconeogenesis? Gluconeogenesis happens in two tissues. One is the liver predominantly. Other is the kidney cortex. Remember that. Although kidney cortex do not contribute to blood glucose level, but the liver gluconeogenesis is the source for blood glucose after 12 hours, 24 hours and then moving on in the first week and then it continues. Anyway, so what are the sources for gluconeogenesis? So the primary sources for gluconeogenesis is alanine coming from skeletal muscle in the first week or any protein degradation going on in our body. In the liver, where alanine coming in there and undergoing gluconeogenesis. Then the lactate coming from anaerobic glycolysis. And then the glycerol coming from adipose tissue triacyl glycerol breakdown. And then it is the propionyl CoA coming from oxidation of branched chain fatty acid or bile acid synthesis. So things like that, or degradation of valine isoleucine, threonine and methionine. So these are the sources of propionyl CoA there. And also phytanic acid oxidation. The enzyme that is primarily involved in gluconeogenesis process, and it is one of the most regulated and controlling point for gluconeogenesis, primarily gluconeogenesis coming from alanine and lactate going into glucose. So that is pyruvate carboxylase enzyme. Now the pyruvate carboxylase enzyme, it is going to convert a 3-carbon pyruvate into 4-carbon oxaloacetate, and that is done by carboxylation process where carbon dioxide is entering into the reaction. So it is a carboxylation process. So pyruvate carboxylase belongs to ligase class of enzyme. Remember that. So it's a irreversible reaction where pyruvate entering into the mitochondria converted to oxaloacetate, because pyruvate carboxylase is a mitochondrial enzyme. It is located in the mitochondria. That information was there in the case stem. Okay. So how do you get pyruvate under fasting and starvation? So alanine is converted to pyruvate by alanine transaminase enzyme. So once alanine is converted to pyruvate, so pyruvate is going into oxaloacetate form. How do you get alanine? Alanine you get it from protein degradation. So initially it is the protein turnover that is going on. Later when the fasting goes like more than 48 hours and all that, so it is the skeletal muscle that is undergoing degradation simply because of lack of insulin and more of cortisol and all that. Anyway, so other source of pyruvate can be lactate that is converted into pyruvate by lactate dehydrogenase. So alanine to pyruvate, lactate to pyruvate. Once you get pyruvate, pyruvate goes down into the matrix of mitochondria, converted to oxaloacetate by pyruvate carboxylase enzyme. So what are the requirements for pyruvate carboxylase? It needs ATP, it needs biotin, and it needs magnesium. And most importantly, it needs acetyl-CoA. Acetyl-CoA is a positive modulator of pyruvate carboxylase enzyme. Remember that. And also remember, acetyl-CoA is an obligate activator of pyruvate carboxylase enzyme. There are two enzymes in our body that needs obligate activators. One is pyruvate carboxylase, this enzyme. Other is carbamyl phosphate synthetase number 1. And the obligate activator for carbamyl phosphate synthetase 1 is N-acetyl glutamate, and carbamyl phosphate synthetase 1, it is there in the urea cycle, and it is the most regulated enzyme in the urea cycle. Remember that point. So these are the two enzymes that needs obligate activators in our body, and in our question it is the pyruvate carboxylase enzyme which needs obligate activator, that is acetyl-CoA. That's why the answer to the question is acetyl-CoA. Say other choices, as you see here, other choices, lactate. Lactate, it is indirectly substrate for pyruvate carboxylase. Alanine, indirectly it is a substrate for pyruvate carboxylase. They are not activators. So, and then NAD. NAD is not really an activator there. So acetyl-CoA is our answer. Coming to ATP. ATP is also one of the requirement for pyruvate carboxylase enzyme. Then why it is not an activator? Although ATP can be, it is a requirement, it's an activator, but the most important activator of pyruvate carboxylase, an obligate activator, is acetyl-CoA. Without acetyl-CoA, pyruvate carboxylase cannot work. Remember that. And it is one of the most important enzyme for gluconeogenesis in the very first week of fasting and starvation. That's why the answer to this question, although ATP can be the answer, but the most important answer is acetyl-CoA. So the questions where the distractor can be inserted in the choice. So ATP is a distractor here, so that you got to go for the best answer, and the best answer for this question is acetyl-CoA, not ATP, although ATP can be one of the answer, but you got to choose one best, and that is acetyl-CoA. I hope this is clear. So let's get into little more onto this enzyme. So alanine to pyruvate, lactate to pyruvate, pyruvate to oxaloacetate, and this oxaloacetate is converted to malate. Malate will move out of mitochondria into the cytoplasm, converted back into oxaloacetate. Oxaloacetate is converted into phosphonyl pyruvate. That is done by phosphonyl pyruvate carboxykinase enzyme, and that needs a GTP, and it's a decarboxylation reaction. So you got 3-carbon phosphonyl pyruvate. So GTP, one of the source for GTP is succinate thiokinase, which is there in the TCA cycle. Remember that. So phosphonyl pyruvate going into all the way to fructose 1,6-bisphosphate, and that is going into fructose 6-phosphate, glucose 6-phosphate, and glucose 6-phosphate going into glucose formation. This is how alanine, 2 alanines converted to 2 pyruvate going all the way back to glucose, or 2 lactate into 2 pyruvate going all the way back to glucose. So in this process, pyruvate carboxylase is the very first enzyme, and later, so we have phosphonyl pyruvate carboxykinase, our second enzyme in gluconeogenesis, and then we have fructose 1,6-bisphosphatase enzyme, third important enzyme in gluconeogenesis, and then glucose 6-phosphatase enzyme, and that is the fourth enzyme in gluconeogenesis. I have a video on gluconeogenesis. Please watch that video. Link for that video is there in the description below. And also, what if there is a deficiency of pyruvate carboxylase enzyme gene, that is the PC gene? So that leads to pyruvate carboxylase deficiency, and the video for that also there in the link for that is there in the description below, and also it is appearing in the upper right corner right now. So, and pyruvate carboxylase, it's an anaplerotic reaction. That means by converting pyruvate into oxaloacetate, it can go into TCA cycle and continue the TCA cycle, because oxaloacetate can be diverted into malate formation to go into gluconeogenesis, or oxalostate can go into aspartate formation in transamination reaction to make aspartate. So all that connections are there. So I have a video on that, so please watch that video. So this is all about the case-based learning using this particular case stem, where I have covered pyruvate carboxylase concept. So very important concept. You got to know about this enzyme. Details in and around that enzyme you got to know really well. That's about it. I'll come up with the next case-based learning. Till then, you take care.